Okay. So this is my disclaimer slide. So in the next 30 to 40 minutes, I will discuss some of the historical background about HLA discovery. Then I will explain briefly how to identify novel HLA alleles using the TSV software from One Lambda. And lastly, I will wrap up my presentation by showing all the necessary steps to submit a novel HLA allele to the IMGT website. So we'll start with some of the historical background about the HLA discovery. So our field started in 1958 when Dr. Dusay noticed that mixing patient serum with another individual cells, it agglutinate. He did two more observations. The patient serum agglutinates some of the leukocyte and some not. And the second observation, he noticed that it never agglutinates the patient's own cells. Dr. Dusay named the antigen agglutinated by these sera as MAC or MAC, and this antigen is now known as the HLA-A2. 10 years later, an official committee was formed, and they published a report which listed all the 8 recognized antigens at that time. And in 1971, anti-sera were identified which only reacted with mutually exclusive subsets of HLA-A9 positive cells. This observation resulted in naming 2 new HLA antigen, the HLA-23 and the HLA-24. HLA-A9 was considered a parent antigen, and HLA-23 and HLA-24 were referred to as the split of HLA-9. In 1980, HLA-B7 was the first gene to be sequenced by chemical degradation. And in 1991, Sanger sequence was introduced clinically to the HLA lab and was the gold standard for high-resolution typing till it was replaced by next-generation sequence in 2010. So in 2010 was also the birth of the new HLA nomenclature, which allow adding infinity number of new alleles. And it's also known as the 4-field resolution. So the first field corresponds to the serological antigen or the allele family. The second field represents the subtype of the antigen, which is typed by the molecular method. And a change in the coding sequence in the DNA that results in a synonymous mutation is represented in the third field. And the fourth field represents difference in non-coding region. Lastly, the suffix at the end represents the expression level. So it could be low, null, or secreted antigen, or questionable antigen as well. So as you know, the HLA system is the most polymorphic region known in human genome. So to date, there is over 25,000 HLA alleles for class 1, as seen in this light green bar, and over 10,000 HLA class 2, as seen in the dark green bar. And I can see the growth of the number of alleles over these years is reflected to the method of typing that was available. So if we took the first phase as an example, the CDC was the gold standard of HLA typing. And in the early 2000, the molecular method started to replace the CDC. And in the third phase, represent the introduction of NGS typing method. And in conclusion, this means that every time there is a new method or there is an advancement of new method, there will be faster and more novel alleles will be discovered. And to date now, a lot of the HLA had switched to the sequence-based method for HLA typing, especially after the new NMDP policy, which required the patient and the donor to be typed by sequence-based method. And here, sequence-based method could be Sanger sequence or could be next-generation sequence. So before that policy, our lab were using SSP as the primary method for high-resolution typing. But after this policy, we switched directly to next-generation sequencing without even an intermediate phase of Sanger sequencing. And also the ASHI standard has updated to include that the high-resolution typing must exclude the entire common, intermediate, and well-documented HLA alleles. based on the latest CIWD version 3. And what's the impact in that? The impact is that now the current basic molecular method that most of the lab were using will not be able to rule out all these CIWD. So if you are not aware about how many HLA alleles are added to the CIWD version 3, I will explain it in the next slide. So back again to the polymorphism graph again. So what you see in the y-axis is the total number of HLA alleles, and the x-axis represent the number of HLA alleles added every year, and it's cumulative. You can appreciate that every year more HLA alleles are added. However, the majority of these alleles are very rare. This means that you might not see them again in your entire life. And so in 2012, a group of researchers established the common and well-documented HLA alleles based on the allele frequency. And they found that only 17% of these alleles are only CWD. And then with advancement of the method, new alleles got added. For that reason, CWD list got revisited again in 2020. And the number of the CIWD HLA were tripled. And now it's called the CIWD version 3. So if anyone asks me why it's important to report a novel HLA alleles, I can simply answer this in 2 sentences. The first is HLA is the most polymorphic region, so it's going to evolve, and there is more genes that will be discovered, even with more if now the NGS is the most current method for typing, but if there's, I don't know, maybe in the next 5 or 10 years, there will be another method, there will be more alleles to be discovered as well, and it'll be easier for the labs. The other thing is, as you have seen, that the CWD has been updated, so the rare alleles that you discover today can be a common HLA alleles in the future. So now let's see how can we identify a novel HLA allele using the TypeStream Visual NGS software. So I'm only focusing on the novel alleles identified by the TSV. So if you are familiar with the TSV or the TypeStream Visual software from One Lambda, the default configuration of health metrics is to red flag any mismatch in any exon. So the first thing I usually examine in any alleles is the health metrics column. So if there is a red flag, I look next at the mismatch column and detect if there is any mismatch in the key or the non-key exons. And the key and the non-key exons, so for class 2, the key exon will be class 2, and the non-key exon will be anything except class 2. So in this example, there is a mismatch in the non-key exon in the DRB345 locus, which could be anything except exon 2. So the next step I look is to look at the read coverage. And in order to do that, I usually click on the DRB345 locus. So when you click on the DRB345 locus, this will open another window, which is the read coverage display. And in the read coverage, if you click on the mismatch allele and then click in the right arrow, it will move you between the base pair till you find the mismatched allele in the exon. And in this example, the mismatched base pair is found in exon 4, which is a non-key exon. The mismatched base will be highlighted in red, as you can see. And the consensus is A, but you need to have G to call it the DRB3020201. So from this simple screen, you can basically think that there is a novel allele that detected by TSV. But this steps is not enough. You need to do extra steps to confirm that this allele is a novel allele. So let's see how can we do it. So now let's get some details about our novel allele. So in this example, you can see the closest allele is the DRB302020102. And there is a mismatch in exon 4 at the position of 659. And you can see that G has changed to A. So the next step now is to click on the info tab, which I highlighted here. And you can see that the genomic position is 11,298. And I actually like this feature in TSV because the genomic position actually match what's in the IMGT database. The codon has a mutation is 191.2, which means that the mutation is present in the second base pair of the codon. And then I look at the amino acid change. The reference allele is CGC, which codes for arginine, but the consensus is CAG, which codes for glutamine. This means that it's a non-silent or a non-synonymous mutation. The most important is to look at the read depth. And in our case, you can see that we have a read depth of 2086, which is like a big sufficient of reads. And you can see that for the depth in A is 380, and the depth for the other alleles is 1590. So there is a slight allelic imbalance. But you have sufficient read in your minor allele to confirm that this is a novel allele. So if we look at the base variation chart, especially at exon 4, and exon 4 here is represented by this purple column, you can see there is some sort of allelic imbalance. So allele 1 in green and allele 2 in blue, but it's well separated from the background noise, which is in red. So this means that we actually have a novel allele. So we confirm it. We did our extra homework. We look at the read coverage. So let's see now how can we describe this novel allele. So in order to describe the mutation, you need to understand the structure of the HLA class 2, which part of the beta chain is transcribed by exon 4. And you can see that exon 4 transcribe the transmembrane protein. So the way you can describe this novel allele will be, as I put it in the slide here. So DR beta 302 is a possible variant of the DR beta 30202 due to the genomic position 11,298 within exon 4 and codon 191.2. This results in a non-synonymous mutation because arginine has become glutamine in the transmembrane protein of the beta chain. And this is actually the non-antigenic recognition domain. And this description is very important because you need to add this description when you submit your IMGT. If you forgot to add it, then this will delay your submission, and they will ask you in a separate email to ask us to submit them the description of this new allele. So I'm going back now to the read coverage again. So in the read coverage, we'll click on the novel bases, and which is in the read window. And you will notice that the software named the novel allele as DR beta 302 new. The next step is to check mark the novel allele and then click on create report. So this actually is an excellent feature in the TSV software because in the past, without this software, it takes a lot of time to create the FASTA Q file, the FASTA files and the table annotation table. But by one click button, you can get them in a second, and we will see how. So after you click create report, this will pop up another window. And you need to find a suitable location in your PC to save these files. It's important to remember where you save these files. And then you type the suitable name for the file. Example, you could use your accession number and then click save. After you save, you'll find two files being created. One of them is the FASTA file and the other is the table file. And most important is, again, to remember the location of this file because it happened to me several times after I saved them, I forgot where I saved them. And I have to go back to TSV and create them again. So if you are curious on how does the FASTA Q files look like, you can easily peek on the file. by opening the file using the notepad. And this is how the FASTA file will look like. In the top, you will see the description of the allele, followed by the whole genome sequence. If you open the feature table file, also through Notepad, the feature table will look like this. This format will allows different kinds of feature to be annotated. So example, you can add the gene, add the coding region, and also qualifier as the product or the node. And you don't need to do anything. The software can do it for you. And it's also restricted to those approved by the International Nucleotide Sequence Database Collaboration. And it's actually a 5-column, and each column is separated by a tab. And that's why it's called a tab delimited table. You can do this table manually if you don't have this feature in your NGS analysis software. But I'm not going through these steps in this presentation, but I added a link here that can guide you on how to do this table manually. So the next step is to confirm that this allele is actual and novel allele. Because remember that the vendor software don't usually run the latest IMGT database. So the IMGT database is updated 4 times a year, but the vendor software IMGT database is usually updated twice a year. So there is usually a lag between the IMGT and the vendor database. So because of that, you need to blast the sequence of your novel allele using this link. And this will open the BLAST website. And from the from the BLAST website, you can click on nucleotide BLAST, and this will open another window. And this is the window that will open after you click the BLAST, and you will see that the BLAST in is selected by default. So the next step is click on Choose File. And again, you have to upload the FASTA file that you saved on your computer. That's why I was asking you to remember where you have saved the FASTA file. And then after you select it, you press open, and then the FASTA file will be uploaded automatically to the BLAST website. So if you didn't do this step, or your software that you're using doesn't have the feature to create the FASTA file, then you have to do it manually by pasting the sequence in the box above in the top left corner of this slide. After you do this, you just click BLAST. And depend on your internet speed and the business of the server, it usually takes a couple seconds before you can get some data. So if you look at the bottom of the screen, you will see hundreds of allele that are very close to the allele that you have just submitted. And by default, it's usually sorted that the most closest allele is in the top, and the least closest allele is in the bottom. So I usually like to choose the most closest allele, because this is what we want to see if our allele is a novel allele or someone else has submitted this allele to the database. So in order to do that, I just click the first allele, and this will open a new window. And from this will open, and from this new window, you can see the sequence that you selected will be compared to the sequence that you have uploaded. So on the top nucleotides are the sequence that was submitted, and the bottom nucleotide are the reference sequence that was just selected. And these lines are the alignment lines. So if you find a line between the two databases, the reference and the one you submitted, you know that there is a consensus between both of them. If there is a missing line, this means that there is a mutation or a new novelty in this mismatch. But actually, I found when you're looking at 11,000 base pair and you scroll, I found that your eye become blind and you wouldn't see if there is a line is missing. So I found an easy way to go around this. So let's see how can we do it. So from the select alignment tab, and then there will be a drop-down menu. you choose the pairwise with dots for identities. And when you do this, I found that it's much easier to view them. And let's see how this will change the view. So this will just change the view of the consensus. So instead of trying to track all the alignment lines, it will be replaced by dots. The presence of dots mean they are consensus. But now if you scroll to the bottom of the page or until you find the mismatch allele, so let's scroll to the bottom of this page. Now it's this is the nice feature I like, because now your mismatch will be highlighted in red. So even if you're not able to track all these dots, but I'm pretty sure you can see the color, and it will be spot that there is a mutation. And you can see here that the mutation is telling you that the G become an A. So now the next step you want to double check is if this is the same mutation that the TSV software has confirmed it for you. And if this allele has been submitted before, you will not find any mismatch when you perform the blast. So now we confirmed actually that our allele is our novel allele is actually a novel allele. No one has submitted this allele before. So now let's go into the gene bank through Bankit and see how can we submit it to get a new accession number. So in order to do that, you have to follow the link that I'm sharing with you here. And this link will open directly to the Bankit website. And after you open the link, you will find a checklist of items that you need to do before submission. If you followed all the previous steps that I just discussed, then you can click submit. And after clicking submit, a new window will open and you need to log in through a third party. Like sometimes I found it easier to use your Google account, or if you have an ORCID account, you can use it, or a Microsoft. But if you don't have an account, you need to sign up. Now, the next step is to click on Start Bankit Submission, which is at the bottom of the next page. The first you need to do is to fill your contact information. And this information is important so that this novel allele would be linked to your name, or linked to your lab, or linked to your place. I fill as much information as I need here. There is no restriction. And then you click continue. The next tab will be the reference tab. You can type your name and as much as author as you need by clicking add button. For publication status, select unpublished because you haven't published this allele yet. And for reference author, I usually select as the sequence author. And then I click continue to move to the next step. So now the next step is the sequence technology tab. First, select your sequence technology. In our case, it's Illumina. If you're using the Ion Torrent, you can pick Ion Torrent. If you're using the PacBio, if you're using a Sanger sequence, you choose any sequence method that you have used. And you need to add, select the assembled sequence, because if you're using a next generation sequence, then it's going to be assembled by a program. In our case, the TSV program is the assembly program, and you need to add the version of the software that you are using. So if you are in version 3, then you have to put version 3. And if you used a third-party event, another assembly program, you can add another assembly program as you need. And then you press continue. In the nucleotide tab, it will ask you about the submission release date. I usually like to select immediately because I want to make sure that that this allele got an accession number. And if anyone else, say in a week or 2 weeks, found the same novel allele, they will know that someone else already have claimed this allele. Select from this window, you will select genomic DNA for molecular type and linear for the topology. And since you are not submitting a virus or a plasmid, then you will select no. In the next nucleotide sequence format, select FASTA sequence. Then next, you will choose file and select the file. And then from here, again, you have to go back to where you have stored your FASTA file and choose this file. And in the submission category, you will select original, because you are the original submitter, and then click continue. And in the source modifier, there is nothing need to be filled, so you can just click continue. You don't need to fill anything here. And if you notice here at the bottom of the slide, you can see that the FASTA files has been uploaded, because you can see that there is a sequence ID, which is 221336, and this is actually our accession number that we used in the FASTA file. So this is to confirm to you that your FASTA file has been uploaded. And then the feature tab, I think this is the most important tab, and this is a lot of the issues, or a lot of the people who submit novel alleles have multiple issues. But if you follow the steps that I have mentioned, you shouldn't have any issue. So first, select add features by uploading a 5-column feature, because we already created this feature table using the TSV, so you don't need to do it manually. And then next, you will choose the file again from the same location that you saved it originally by the TSV. And then click open, and then click upload file, and this will upload your FASTA, the table format file. And then after that, you will click continue. Now in the feature tab, you will see the annotation table that you just uploaded at the bottom of the screen. And in some occasion, you will see a warning like that. And I only have seen this recently when we submitted the last couple alleles. I'm not familiar what this warning is, but I found it's not affecting your submission, and I haven't got any email back so far from the staff from the gene bank. So it could be some sort of just want to make or want to confirm that you have a name assigned to this novel allele. The last tab will be your review and correct. You can scroll through the whole page, and if you find any mistake, you can go back to any tab to fix it. And after finishing the reviewing, you can just hit finish submission. And after you've clicked finish submission, you will get a confirmation message, which will show up and your submission number will appear. And it's important to keep this number, because if, for example, if there is a delay happen in getting your accession number, you will have this submission number so that you can track your records, or you can email them, ask them about your update of your novel allele. So please write this number after you click the submission. And I usually found it takes about 48 hours. They are really fast in that. And then you will get an email from the Bankit staff, and then they will give you the accession number. And I found that this accession number is formed of 3 different numbers. The first part is your submission number, which is matching actually the same number here. And then the last number is called, I think this is called the accession number, because this is the number the IMGT needs, which is usually start by a letter followed by a number. And then in the center here, this is the sequence ID. And this is the name of the file that you named your FASTA file with. And in our case, it was just the accession number. But if you want to ignore the first 2 numbers, or the first 2 parts of this number, you need to remember and write up the last accession number, which starts with OP and then a bunch of numbers, because this is the number that you are going to use for the IMGT database. So now after you receive the accession number from the gene bank, you are ready to submit the sequence of this novel allele to the IMGT website. So open the IMGT submission site by clicking on the following link and click on Start Submission. Now you can read through the whole checklist and make sure you fulfill all the criteria of submission. And if you fulfill all the criteria of submission, you can please check all this mark. And I just want to remind you about a very important item here is if you submitted a DNA from a hematological malignancy patient, you need to confirm this by also a buccal swab, because we want to exclude that there is a somatic mutation, and all what you are sending is a somatic mutation of the cancer cells, not actually a novel allele. So if you are doing DNA from peripheral blood from hematological malignancy, before submitting it to the website, is to do it through a buccal swab. After that, you press proceed to next step. And then in the next step, you will find you need to fill again all your contact information, because this information will be linked to the name that the IMGT will give you. So it's nice to track all your novel alleles by tracking your name under these novel alleles. If you don't want to add your name, you can add the name of your lab as well. And then in stage 3, which is the basic sequence information, you need to give a local name to your new allele. For example, in our case, we called it DR beta 302 new. The sequence type is it's a class 2 genomic. So from the drop-down menu, you choose a class 2 genomic. The accession number is the number you have received from the gene bank, which is formed of 3 parts. You only need the last part, as I mentioned before. So don't add the full number, because if you add the full number, this will delay the submission, because they will double check with you in a separate email. And then the release date, again, I usually put the release date as the date of my submission. So because I want them to be accessible as fast as it can. After that, you can press continue. And then from the submission reference details, since it's not published yet, you will choose unpublished data, and from the drop-down menu, and then you can click continue. You don't need to fill anything here. And in stage 5, which is submit nucleotide sequence and the sequence feature, you need to select the locus, and in our case, it's the DR beta locus. Then choose the closest allele, or actually you have to type it, and you have to type the closest allele in a four field resolution. So here you can see that the closest allele that I typed is the DR beta 3, 02, 02, 01, 02. In the box, in this box, which is the number 3, this is where you have to put a detailed information about your novel allele. And if you miss this, they will start emailing you, asking you to put a detailed information. And now we will go back to our FASTA files. And from the FASTA files, you can open it through the notepad, but now you need to copy it manually. And in order to copy it manually, you need to select from the first nucleotide base. You don't need to put the title like the one you see, the title, which is the first 2 sentences. You don't need to copy it. So your first copy will be the first nucleotide, and then the last thing to select will be your last nucleotide. And then you'll do right click and copy, and then you will paste it on where it says nucleotide sequence feature. And then from the drop-down menu, you will choose assisted assignment of feature. And then the number of features, in our case, it was 11. because we are submitting 6 exons and 5 introns. So if you are submitting less than that, then you have to put the total number of the features that you are submitting. And then click continue. Now the feature table will be populated. The software will create this feature table to you, which shows you the start and the end of for each exon and intron. And because you selected that you need the software to do it for you, the software will automatically do it for you based on the sequence that you added, and the gene that you have chosen, and the number of annotation that you have added. And in the next step, you can now scroll down to the page. You can see the full summary, which include all the feature and the sequence of submission. And in stage 7, which is the information on the source individual, you can fill as much information as you can, as this will help other labs that find the same allele to get an idea about the ethnicity of the individual. And also, if you added the full HLA typing of the individual, other labs that identify the same allele can look for the haplotype similarity. And I believe that the minimum that you need to add is locus A, B, and DR. But if you add the more will be better. So I will take here an example on how to do it. So for each gene, you need to add the 2 alleles in the same box, separated by a semicolon. So if we take HLA as an example, you can see I added 0101, semicolon 1101, and so on. You have to continue in all these alleles. So in stage 9, talks about the method. From the drop-down menu, you select your primary and the secondary sequenced method. In our cases, it's both the primary and the secondary are Illumina sequence. So you need to, and then you need to confirm that the allele has been sequenced in isolation from the second allele. And since you are doing NGS, then you can confirm its sequence in isolation. And then now you scroll down to the rest of the page. And no, you don't need to add the sequence of the primer because I don't think you will know them. So you can leave them blank. And in the sequence direction, please select both because most NGS assay sequence both forward and reverse strand. And you can choose the confirmation method. We usually confirm novel alleles by another vendor NGS. You don't have to, but we found that this is more clinically relevant for us. And then you can click, you can hit the submit button. So you will get a confirmation message that your submission is complete, and you will get a submission number. Again, this is the number that you can use to track your submission to the IMGT. And the staff of the IMGT are very helpful. They can guide you step by step if you have any questions as well. And I think I found it usually takes between 4 to 6 weeks before you get an email. So please be patient. Because they are so busy assigning all these new alleles. And I believe the committee usually meet once a month, and they submit this novel allele. So wait around 4 to 6 weeks, and then you will get an email with a PDF with the name of your new allele. And in our case, it was the DR beta 302 185. And you can see that we got this letter in September 30, and we actually submitted this in September 9. So because everything was smooth, they didn't have to go back and to us to ask a question. So the submission went very smoothly and quickly in a couple of weeks. Now it's time to document your hard work in a sort of publication. So the most common journal that accepts novel allele alert is the HLA journal. Submission is free. So here is the link of the journal. So first you need to log in or create an account, and then click submit an article. Then you hit start submission. Now select the article type that you need to submit. And then from the drop-down menu, you need to select New Allele Alert from the drop-down menu. And then you need to check all the checklist. And then scroll down and hit confirm. You will now, if your manuscript is ready, you can upload your manuscript. And the manuscript you don't have to be like a very it's more like a one-page summary about what did you find and what's the method that you used and how this affects the expression of this HLA allele. And I usually write it in a one-page summary. And you don't need to put a lot of references. I think the maximum references you add is like 5 references, and also you have a maximum number of authors. I think it's around also 5 authors. So it shouldn't take you a very long time to write it. And if you have a subscription to the HLA journal, you can review example of this novel HLA allele report. And then you will click that there is no conflict of interest, and then follow the rest of the guidelines of the new allele alert. And this is an example of an HLA allele report that we published, and the link of this manuscript is at the bottom if you're interested in seeing how it looks like. And then you need to remember, or the take-home message of this is, you need to do 4 important steps before submission of any allele. So the first thing is, and I found this is the most important, is to do a nucleotide blast, because if you didn't do a nucleotide blast and then after you spend all this time, then you will get a response from the IMGT staff that your allele has already been submitted. So please do the blast as the first step. The second step is to submit the novel HLA allele sequence through the gene bank, through the Bankit, and then submit the novel allele through the IMGT HLA website, and document this hard work by publishing it into the HLA journal. And I would like to thank at the end of my the staff at the HIL lab in Saskatoon at St. Paul Hospital for their hard work. And I'm actually only here to present this work, but these are the right people, or the people behind the scene that do all this extra excellent work. And I also like to thank One Lambda for facilitating this webinar. And I can stop now my screen, and I'm ready to answer all your questions.